Hello children, in the last class we have studied about pleiotropy gene. That means one gene is responsible for more than one character, right? Now there we have studied sickle cell anemia. Due to mutation of one gene that causes a change in the hemoglobin, and this also caused a change in the red blood cell. Okay, that patient which is suffering from sickle cell anemia, they can able to survive in the malaria prone area, right? This is what we have studied in the last class. Here in this class we are going to study about polygenic inheritance, that is, it is otherwise known as quantitative inheritance. Quantitative inheritance means it is the quantity of the gene which determine the character. That means more than one gene is responsible for one character. Till now students, what we have learned, one gene is responsible for one character, and pleiotropy what we have learned, that is one gene is responsible for more than one character. Here in polygenic inheritance, more than one gene, okay, is responsible for one character. Example, here we are going to take the example of human skin color. Example of skin color in human. You can see a range of skin color in human. Okay, this skin color is controlled by 3 genes. It is controlled by 3 genes. That is why it is known as polygenic inheritance. Okay, what are the 3 genes? A, B and C genes are responsible for the skin color. The number of this dominant gene would decide the amount of melanin pigment. Okay, suppose a person is having all the gene in homozygous dominant condition, that is capital A, capital A, capital B, capital B, capital C, capital C. It can able to produce maximum melanin. Okay, they can able to produce maximum melanin. Okay, all the genes are in dominant. So we can count here 1, 2, 3, 4, 5, 6, total 6 are there in dominant condition. So the skin color shows black. The skin color shows black. Now let us see the intermediate, which is in all the genes are in heterozygous condition. What is heterozygous? That means, let us write here, capital A, small a, capital B, small b, and capital C, small c. Okay, how many dominants are there? Here only 3 dominants are there. Here 6 dominants are there, so maximum melanin is synthesized. Here there is only 3 dominant, so medium melanin is synthesized. Medium melanin is synthesized. Okay, so as it produces medium amount of melanin, the skin color will be intermediate. The skin color will be intermediate, or you can say wheatish color. Okay, now other extreme, let us see the other extreme here, in which all the genes are in recessive condition. All the genes are in recessive condition, so there is no dominant gene. So 3 means there is no dominant gene, so we can write here as 0 dominance. Okay, as there is no dominance, no melanin is synthesized. Okay, when there is no melanin, the color of the skin is albino. The color of the skin is albino because there is no pigment, melanin pigment. Now we can see a range from intermediate to towards this dominant and a range from intermediate towards this recessive. As we move from intermediate towards the dominant, the number of the dominant gene increases. Let us see here. That means capital A, small a, capital B, small b, capital C, capital C. So how many dominants are there? 4 dominants are there. A, B, C, C. So 4 dominants are there. Okay. So there is 4 dominants. That means it produces more melanin from this. Okay. So the skin color will be dark. Right. Now as it moves towards this direction, that means it is having capital A, small a, capital B, capital C, capital C. How many dominants are there? 5 dominants are there. Okay, 5 dominants are there, more melanin is synthesized. So the skin color appear very dark. So the skin color appear very dark. Okay, here 6 dominants is there and maximum melanin is synthesized. So the skin color is black. Now as it moves from intermediate to this extreme, we can see the number of the dominants decreases. You see here capital A, capital B, small c, small c. How many dominants is there? Only 2 dominants is there. Okay, so as the number of dominants is decreased, there is a level, decreased level in the melanin pigment. So it appear, the skin color appear fair. Okay, now again the dominant gene is decreased, capital A, capital small b, small c. There is 1 dominant gene. Only 1 dominant is there. So again the melanin synthesis is decreased, become minimum, and the skin color will be very fair. So in the last stage there is no dominance, 0 dominance. So no melanin is synthesized and the skin color is albino. Okay, see, this is the phenotype. Okay, phenotype of the skin color. Okay, what is physical appearance? And this is the genotype. Genotype, that is the genetic makeup. Okay, now let us see a cross in which the 2 extremes are crossed. What are the 2 extremes? That is black and albino is crossed. Let us see that. Black is, let us write here black. We consider this as parents. Okay, black when it is crossed with albino. Black is genotype, all are dominant, all the genes are in dominant condition, that is homozygous, and albino, all the genes are in recessive condition. All the genes are in recessive condition. What is the next step? The next step is gamete, formation of gamete. As we have studied in the principle of segregation, this allele will separate and enter into the gamete. So how many genes are there? There are 3 genes. Okay, from these 3 genes are having two alleles. So during gamete formation, each allele from every gene enter into the gamete. So we can see capital A, capital B and capital C. So this allele, one allele from each gene is entered into the gamete. Similarly here also small a, small b, small c. So what is the F1 generation? Progeny obtained from this cross, can say that an intermediate color is obtained. How? Capital A, when it is fused. Capital A, capital B, small b, capital C, small c, and intermediate color. Intermediate color is obtained. When two extremes are crossed, a black is crossed with albino, what is the progeny obtained in the F1 generation? That is an intermediate color. All the progenies are in intermediate color. When these progenies are breeded, okay, when the two intermediate progenies are breeded, and in the F2 generation, here how many genes are there? Total 3 genes are there. We can say the first, when we take only one character at a time, we call it as a monohybrid. Second, we call it as a dihybrid. Third, it is having trihybrid. Okay, trihybrid, or 3 genes are present. Okay, this cross is known as trihybrid cross. Now, we can see the phenotypic ratio of this F2 generation. Okay, now the phenotypic ratio obtained from this F2 generation ratio is 1:6:15:20:6:1. So, here you can see the ratio, total 64. In the monohybrid, the total number will be 4, and in dihybrid it is 16, and in trihybrid it is 64. Now, this is the extreme. What is this? That is dark, or you can say it is black, and here it is albino, and these two are very dark, and this is very fair. This is dark, and this is fair, and this one is intermediate. Okay, so there are total 64 genotypic probability, that is possible genotype in the F2 generation is total 64. Okay, it is in the ratio of 1:15:20:16:1. So we can say 1 black and albino, 6 very fair and very dark, 15 dark and very that is fair, and the intermediate is 20. Let us see this. We can plot this ratio in a graph. So this is a graph formed by the ratio of F2 phenotypic ratio, that is the phenotypic ratio of F2 generation. So these are the possible genotype. Okay, these are the possible phenotype. How many genotypes are there in this condition? Here, that is, all are in homozygous, only one is there. Here all are in this is homozygous dominant, here it is homozygous recessive. One possibility is there, and the color is here black, and here it is albino. The next possibility is one is recessive, all the remaining one are dominant. So you will get 6 possibilities. Here one is dominant and the remaining are recessive, that is also of 6 possibility. And here it appear dark in color, here it appear very fair. This is very dark and very fair due to the presence of 5 dominant gene. Here due to the presence of 1 dominant gene it appear fair. Now this is the possibility, that is 15 number of genotype, there is a possibility of 15 number of genotype. So how many dominants are there? 1, 2, 3, 4. Here only 2 dominants are there. The color is dark and here the color is fair. There is a possibility of 20 genotypic possibilities are there, which shows intermediate color, in which 3 dominants are there. So we are getting a bell-shaped graph. So this is about polygenic inheritance. So what is polygenic inheritance? That is the more than one gene is responsible for one character. Okay, how many genes are here? We have taken the example of skin color. The skin color is due to 3 genes, that is A, B, C. Okay, the amount of dominant gene would decide the amount of melanin pigment. Is it clear? Okay. Next we will study about gene interaction. What we have studied in principle of independent assortment, when two traits are taken for a cross, they separate or segregate independently and they can express independently. Okay, we have taken the colors of seed and shape of seed. Both the genes are not interfering. Okay, that is a type of dihybrid cross. Now here you can see we are taking 2 genes or 2 characters. Genes means which carries the character. Here one gene is interfering with the other gene. Okay, so one of the example is complementary gene. Complementary gene. Example of complementary gene is this, we can see in sweet pea. Sweet pea. Scientific name of sweet pea is Lathyrus odoratus. Here flower color is one trait. This flower color is interacted by 2 genes. The expression of this flower color is due to the presence of 2 genes. So let us try to understand. Actually the color of the flower is purple and the contrasting trait is white. Okay, purple is dominant and white is recessive. Actually what happened in Lathyrus odoratus, there is a raw material in its body. In plant's body raw material is there. This raw material is converted into chromatin by an enzyme E1. Okay, this enzyme is produced by a gene capital C. This is a gene capital C. This gene is responsible for the production of enzyme E1. Okay, now this chromatin is converted into anthocyanin, which is purple in color, that gives a purple color to the flower. For this conversion, that is from chromatin to anthocyanin, what is required? There is an enzyme which is called the E2, and for the formation of this enzyme, a gene is responsible, that is known as the P gene. Okay, so both the C and P are in dominant condition. Suppose this gene which is in a dominant condition, if it exists in recessive, what will happen? This in recessive form, it is unable to synthesize enzyme E2. As it is unable to synthesize enzyme E2, chromatin will not be converted into anthocyanin, so the color, flower of the color will be white, even though if it is in a capital form. Okay, so if it is in a dominant condition, if it is in a recessive condition, then also the color of the flower will be white. In other case, we will take here, it is in recessive form. Suppose this gene is in recessive form and this gene is in dominant form. What will happen? As this gene is in recessive form, it is unable to produce enzyme E1. So as it is unable to produce enzyme E1, so raw material will not be converted into chromatin. Okay. Now, if raw material is not converted into chromatin, even though this gene is in dominant condition, purple color will not appear. So what you have learned from this? That means this gene complement the other gene, or that gene complements, both the genes are complementing each other. Okay, the purple color appear when both the genes are in a dominant condition. So let us see a cross. Here we are taking the cross. We consider this as a parent. Two white plants are taken for the cross. White is crossed with white plant, white flower. Two white flower producing plants are crossed. Phenotypically they appear white. Genotypically they are capital C, capital small p, small p. This gene is dominant, but this gene is recessive. That is why the color of the flower is white. Here the genotype of this flower is small c, capital P. This gene is in capital or dominant, and this gene is in recessive, then also the color will not appear. Okay. Now next step is gamete formation. How the gamete formation takes place? One allele from this gene and one allele from this gene enter into the gamete. Okay. And one allele from this gene and another allele from this gene will enter into the gamete. Okay. When this gamete are fused, we will get the F1 generation. So we will be getting an F1 generation, that is capital C, small c, capital P, small p. But what will be the color of this flower? As both the genes are in dominant condition, one of this allele is dominant and second genes, one of the allele is dominant. Both are complementing each other, so the color of the flower will be purple. Okay. So when we take the F2 generation, phenotypic ratio of F2 generation, or next step is the gamete formation. So how the gamete is formed? By independent assortment, capital C and capital B go togetherly, capital C and small p will be present in one gamete, small c, capital P and small c, small p. So suppose it is a male gamete, similarly a female gamete is formed. So let us see the Punnett square. So this is the Punnett square of F2 generation. Now you see here, when we crossed or when we fused all these gametes, we will see the ratio. How many purples are there? 1, 2, 3, 4, 5, 6, 7, 8, 9 purple. Okay, 9 purple. And how many whites are there? 1, 2, 3, 4, 5, 6, 7 white. 7 white. So what is the phenotypic ratio? Phenotypic ratio is 9:7. So what is the phenotypic ratio of dihybrid cross? We have studied in independent assortment. 9:3:1. Okay, this 3 get joined together and here it formed a different ratio, that is 9:7. So there is a deviation from the principle of independent assortment. Likewise, so many gene interaction is there, supplementary gene is there. In supplementary gene, the phenotypic ratio is 9:4:3. Okay, so actually gene interaction is not there in your syllabus, but the aspirants of NEET, you have to remember all the gene interaction. There is supplementary gene is there, and complementary gene, epistasis dominant, epistasis recessive, and duplicate gene. You have to remember the gene interaction along with the examples and ratio. So what you have learned from here? That means there is a deviation from the principle of independent assortment. Is it clear? Now what we have learned till now, that the principle of independent assortment, the principle of inheritance consists of 3 principles. What are the 3 principles? That is principle of dominance, principle of segregation and principle of independent assortment. Principle of dominance is deviated. Why? Because of the presence of codominance and incomplete dominance. What is codominance? One of the allele, that means both the allele of a gene togetherly expresses. Example of AB blood group. In incomplete dominance, what we have seen, neither of the allele is completely expresses. That is, they are unable to express completely, one allele is unable to express completely over the other allele. Example we have seen, that is Mirabilis gelapa, Antirrhinum majus. Okay, here there also the phenotypic ratio is changed. That is phenotypic ratio from 3:1 in dominant, according to principle of dominance, the phenotypic ratio is 3:1. There it get deviated and the ratio become 1:2:1. Again, in the principle of independent assortment, there is a deviation due to gene interaction. So what you have seen, the principle of inheritance deviated, showing some other character. In the population, different characters are appeared. That is why there is a deviation. But the principle of segregation remain as such. There is no change in the principle of segregation. What does principle of segregation say? Pair of a allele means pair of a gene is having a pair of allele that will segregate or separate during gamete formation, and the gamete will receive only one allele of a gene. So the principle of segregation is known as the universally accepted principle. You will get a question. Name the principle of inheritance which is universally accepted. Which principle is universally accepted? That is principle of segregation. Why? The principle of segregation is followed during the gamete formation. So for every sexually reproducing organism, what is required? Formation of gamete is required. So that they follows principle of segregation. What is principle of segregation? Pair of allele segregate during gamete formation and the gamete will receive one allele of a gene. Okay, but the principle of dominance and the principle of independent assortment is deviated due to the certain presence of certain inheritance, like in the principle of dominance is deviated due to the presence of codominance and incomplete dominance. Principle of independent assortment is deviated due to gene interaction. Is it clear, children? So I hope you might have understood the portion very clearly. Okay, we'll see in the next session. Thank you.